Hello everyone, my name is Gary O'Mealy, and in this video we are going to discuss lipids. So lipids are going to be a little bit different compared to some of the other macromolecules that we have looked at and are going to look at, but one of the things that all lipids have in common with each other is that, number one, they are mostly hydrocarbon molecules, meaning that they're mostly going to be made of carbon and hydrogen. And we've seen in a previous video that anytime you have a molecule that relies highly on carbon-hydrogen covalent bonds, that's going to mean that that molecule is going to be very nonpolar, very hydrophobic, and is not going to be very soluble in water. So of the different types of lipids that we're going to talk about, the first one we will talk about are fats or triglyceride molecules. These are going to be kind of most in line with what we've talked about previously with dehydration synthesis and how you typically go about constructing macromolecules. Triglycerides are constructed by dehydration synthesis, but the issue here is that we cannot get unlimited polymerization like we could with carbohydrates or proteins or nucleic acids. So what we can do is we can attach up to 3 fatty acids to a glycerol backbone, but that is all we can do in terms of polymerization here. In the illustration, you can see the glycerol backbone, which contains 3 hydroxyl groups lined up one right after another, and those are going to serve as the attachment sites for the 3 potential fatty acid chains that we are going to attach. When we line up the 3 fatty acids head-to-head with the glycerol molecule, you can see the element of water that will be lost in each case for each dehydration synthesis reaction that occurs. And if we do see all 3 of the dehydration reactions occur, we will attach all 3 fatty acids to the glycerol backbone, and this will yield us a triglyceride molecule. So a triglyceride specific properties are really going to be determined by the type of fatty acids that get attached. Now, in terms of properties, there's not really going to be anything that makes a fatty acid anything other than very nonpolar and very hydrophobic, but we are going to see that there are some different types of fatty acids that we can attach here. Fatty acids can be different in terms of how short or how long they are. They can be as short as just a handful of carbon atoms. They can be very, 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 very long. But most of the important fatty acids in human physiology are going to contain somewhere between 12 and 18 carbon atoms. So fatty acids can come in 2 basic varieties. They can either be saturated or unsaturated. If you've ever looked at the back of a nutrition facts label on your favorite food product, you're probably at least aware of these terms, even if maybe you don't know exactly what they mean. So to illustrate the difference between a saturated and an unsaturated fatty acid, take a look at the fatty acid that's pictured below. You can see the carboxyl group right here that serves as the potential attachment point onto glycerol, and then you see a long hydrocarbon tail. The first thing I want you to notice here is how every carbon in the backbone, with the exception of the carboxyl group, that doesn't count, every carbon atom is completely covered in hydrogen atoms. Keep in mind, every carbon is going to form up to 4 covalent bonds, so the fact that each carbon is participating in the maximum number of covalent bonds, and each bond is formed with the maximum number of hydrogens, this makes this fatty acid a saturated fatty acid. Like I said, you need to remember that every carbon atom is going to want to form 4 covalent bonds. So the issue here, what makes this a saturated fatty acid, is the fact that every single carbon is saturated with the maximum number of hydrogens it possibly can be. But in a different scenario, what if there is a double bond in the carbon-carbon backbone here? You can see the presence of this double bond here. In a double bond, there are going to be 4 electrons being shared between the 2 carbon atoms instead of the typical 2 that you see with a single covalent bond. So, because of the presence of this double bond, that is going to limit the number of hydrogens that can be attached to the backbone. In essence, we are going to remove 2 hydrogens, you can see kind of the vacancy here, in favor of forming this double bond. So these types of fatty acids that contain double bonds in the backbone are called unsaturated fatty acids because the presence of one or even more double bonds prevents the backbone from being saturated with that maximum number of hydrogen atoms. In this particular unsaturated fatty acid that's pictured here, the double bond is in what we call a cis configuration, meaning that the two hydrogens that are on the carbons on either side of the double bond, these two hydrogens are on the same side of the double bond. Cis is a prefix that refers to on the same side versus trans, which means on opposite sides. So this is a double bond in the cis configuration. This is important because unsaturated fatty acids that have a double bond in the cis configuration, the backbone will bend or kink at the double bond. It's not illustrated here because I couldn't really figure out how to do it with PowerPoint, but you'll see an illustration on the next slide that demonstrates this. So in this picture, you can see the difference in molecular geometry between a saturated fatty acid and an unsaturated fatty acid. The saturated fatty acid is a very linear molecule, whereas the unsaturated fatty acid takes up a lot more molecular space because of the presence of this kink in the backbone because of that cis double bond. Like I said, saturated fatty acids and so-called trans fatty acids, where those hydrogens are on opposite sides of the double bonds, they do not have that kink, so those molecules are very linear, and their density tends to be very high because you can fit a lot of these fatty acid molecules in a very small volume. Unsaturated fatty acids have much lower density because these molecules take up a lot more space, and because of this, unsaturated fatty acids tend to be present in lipids that are liquids at room temperature, like oils, like canola oil, olive oil, and things like that. Whereas saturated and trans fatty acids, because of their high density, they tend to be solids at room temperature, things like butter and lard. So as far as functions for triglycerides go, triglycerides are one of the major sources of energy for cells in the body. So as far as how cells go about breaking down triglycerides to get energy through cellular respiration, we're not going to spend a whole lot of time on that right now, but basically fatty acids and glycerol have to be broken apart by hydrolysis, and then they get used separately as fuel sources. Glycerol can be metabolized into a end product of glycolysis, namely pyruvate, and fatty acid can get converted directly into acetyl-CoA, which would then enter the Krebs cycle. The process of oxidizing fatty acids through cellular respiration is called beta oxidation, and it largely occurs in mitochondria and in peroxisomes. Something that is very interesting is that fatty acids actually provide quite a bit more energy per fatty acid molecule than glucose does. So in terms of making ATP molecules, it's a lot more efficient for cells in the body to use fatty acids than to use glucose. But the issue, as we will see in the future as we go throughout this course, is that some tissues are actually very particular about what kind of oxidizable substrate they use for cellular respiration. A good example of this is the brain, which will only use glucose for making ATP and never fatty acids. So we always have to make sure that glucose is available to every cell in the body. Similar to what we saw with excess glucose, which can be stored as glycogen in liver and skeletal muscle, excess fatty acids and triglycerides are stored in adipose tissue as large fat droplets that are stored in cells called adipocytes. And we will look a little bit more at this in Chapter 4. The next type of lipid that we'll talk about are called phospholipids. Phospholipids, as we are going to see, make up a very critical part of the cell membrane, and since our next chapter, Chapter 3, is going to cover a lot of the cell membrane and cell membrane structure and function, we'll have a lot more to say about phospholipids in a future video, but we'll just briefly introduce them now. Phospholipids are very similar in composition to a triglyceride in the sense that phospholipids are made up of a glycerol backbone and attached fatty acids, But the difference is that instead of having 3 fatty acids attached to the glycerol backbone, you only have 2 fatty acids, with the third position on the glycerol backbone being attached to a polar phosphate-containing functional group. This actually causes this diglyceride. It's a diglyceride because there's only 2 fatty acids attached. This diglyceride has a polar hydrophilic end to it. That's the polar phosphate-containing group on one end of the molecule, and then the pair of nonpolar hydrophobic fatty acid tails being on the other end of the molecule. You can see this illustrated here in this phospholipid. You have the glycerol backbone here. Here is your polar hydrophilic head right here. And here's your nonpolar hydrophobic tails over here. Again, this is a diglyceride because you have 2 fatty acid tails attached to the glycerol backbone, and it is not a triglyceride because a triglyceride would have a third fatty acid attached right here instead of the phosphate-containing group right here. So as I said, phospholipids are a major critical component of cell membranes, and we'll have more to say about that in a future video. The next type of lipid I want to talk about are steroids. These are freestanding molecules that are not subject to polymerization through dehydration synthesis, so they're not really going to apply exactly in the same way as what we talked about with triglycerides. A steroid molecule is going to consist of 4 fused hydrocarbon rings, kind of looks like a little bit of a honeycomb shape, so they should be fairly easily identified. And this 4 fused hydrocarbon ring backbone, we can see a variety of different attached functional groups which will give these steroids different properties. The most common kind of steroid in the human body is cholesterol, as you can see here. You can see the 4 fused rings here, and that is going to be something that just about every steroid has. But what you're going to see is that when we talk about other types of steroids, you're going to see a variety of different functional groups that can be attached to basically any of the carbons in the backbone here. Cholesterol itself gets a bad rap sometimes because in the news and in the media you often hear about the dangers of high cholesterol. Well, the reality is that cholesterol is not a bad thing. It is actually a very essential component of our diet and something that is absolutely essential for proper physiological function. Cholesterol itself is obtained, of course, in the diet, and it is also synthesized to a certain extent in the liver. So you absolutely do need to eat foods that have cholesterol in them because, as you're going to see here very soon, cholesterol is very important for a variety of reasons. The first reason is that cholesterol is a major component of the cell membrane. We'll see more about this in Chapter 3 when we actually talk more about the cell membrane, but cholesterol actually wedges itself into the cell membrane, and since cholesterol is a hydrophobic molecule, it wedges itself in the portion of the cell membrane that is hydrophobic in nature, and it helps to increase the membrane's fluidity. More importantly than that is that cholesterol is the basis for all steroid hormone production in the body. This includes steroids you're probably aware of like testosterone, estrogen, and cortisol. All of these hormones are very important for a variety of different reasons. They help to regulate various homeostatic processes in the body by carrying information over long distances from their gland of origin to effector tissues all throughout the body. But what we're going to see here is that all of these hormones are very structurally similar to cholesterol itself. And the body actually saves a lot of time and energy. Rather than synthesizing these steroid hormones literally from scratch, these hormones can be very easily made just by making a handful of chemical modifications to cholesterol itself. In this diagram here, you can actually see an enzyme-catalyzed pathway in which a cholesterol molecule can be converted into an important sex hormone called progesterone. One enzyme will modify this hydrocarbon functional group here and turn it into this more polar functional group right here. And then another enzyme will come in and oxidize this hydroxyl group down here into a ketone functional group. So you can see that when you compare the molecular structures of cholesterol, pregnenolone, and progesterone, they all look very similar to each other. They all have that familiar four-fused ring honeycomb shape. But what makes one steroid different from another steroid is just the presence of functional groups in different places along the ring structure. So these enzymes that you see here that are making these chemical modifications to cholesterol, they tend to be found in the mitochondria and the smooth endoplasmic reticulum of cells that are making these hormones. So these enzymes are going to be catalyzing the chemical reactions that result in the addition, removal, and modification of cholesterol functional groups to make other steroids. So in the future, when we talk about steroid hormone production, you're going to want to keep this in mind. And the last type of lipid I want to talk about are prostaglandins. These are signaling molecules that are derived from unsaturated fatty acids, and they tend to be associated very much with inflammation, which is a usually undesirable process that is the result of tissue damage and injury. So the pro-inflammatory prostaglandins you see below were derived from a type of fatty acid called arachidonic acid. Arachidonic acid is an example of what is called an omega-6 fatty acid. You may have heard of the term omega-6 fatty acid or omega-3 fatty acid before, and basically what you can see here with this prostaglandin is the remnant of what used to be an unsaturated fatty acid. You can see the carboxyl functional group right here, the COOH. You can see a few double bonds here and there. The way that you can determine what type of fatty acid it was is when you find the carbon that is furthest away from the carboxyl functional group, this one right here, that is by default called the omega carbon. If we count the number of carbons away from this, 1, 2, 3, 4, 5, and 6, after 6 carbons we find our first double bond. That is how we determine that this is an omega-6 fatty acid. If this double bond were found 3 positions away rather than 6 from the omega carbon, it would be an omega-3 fatty acid instead. So we can see here that omega-6 fatty acids tend to be associated with the production of pro-inflammatory prostaglandins, which are going to cause redness and swelling and pain at the site of tissue injury. So-called anti-inflammatory prostaglandins, which will alleviate those sorts of symptoms, they tend to be synthesized from omega-3 fatty acids, which are only available through the diet. So if you've ever been advised to eat foods that are rich in omega-3 fatty acids, things like fish oil and krill oil, this is the reason why, because it's thought that these omega-3 fatty acids lead to the production of these anti-inflammatory prostaglandins, which will relieve the symptoms of inflammation. One last little tidbit I want to leave you with here is the mechanism of action for so-called NSAID medications. NSAID stands for non-steroidal anti-inflammatory drugs, things like aspirin and naproxen. And I want to introduce to you, since we're talking about prostaglandins anyway, how these drugs are meant to work. So we talked about how omega-6 fatty acids like arachidonic acid can get converted into inflammatory prostaglandins. The enzymes that catalyze this conversion into prostaglandins is called cyclooxygenase, or COX. One of these COX isoforms, called COX-2, is inducible during tissue damage, and it is thought to be one of the major factors that leads to inflammation at the site of injury. So non-steroidal anti-inflammatory drugs like aspirin inhibit the enzyme, preventing this COX-2 from doing its job, meaning that if the enzyme is inhibited, it will not be able to produce these inflammatory prostaglandins, even if the substrate, arachidonic acid, is available. Okay, so that's going to do it for this video. Here is a list of vocabulary terms you probably ought to be aware of after watching this video. And for checking your understanding, number 1, how do most lipids interact with water? Number 2, what role can lipids play in energy management in the body? Number 3, what important roles do phospholipids play? And number 4, how can lipids participate in cell signaling in the body? So that's going to wrap up this video. If you have any questions, go ahead and drop them in the comments section, and I will see you for our next video. See you next time.